So, hello students, I am here with the almost last topic of this chapter, that is mycorrhizal fungi facilitate nutrient uptake by roots. In this section, we are going to discuss about mycorrhizal association in plants. So, mycorrhiza. Mycorrhiza is a type of fungi which get associated with the roots of large trees. So, this fungi is a type of root fungi which get associated with the root cells and provide them absorbed minerals elements from the soil. So, mycorrhiza are not unusual. In fact, they are widespread under natural conditions. Much of the world's vegetation appears to have roots associated with mycorrhizal fungi. 83% of dicots, 79% of monocots, and all gymnosperm regularly form mycorrhizal association. Few plants like Cruciferae and Chenopodiaceae and Proteaceae as well as few aquatic plants rarely have mycorrhiza. Mycorrhiza also absent in dry, saline and flooded soil or where soil fertility is extreme, either high or low. In particular, plants grown under hydroponic and young, rapidly growing crop plants seldom have mycorrhiza. Mycorrhizal fungi are composed of fine tubular filaments called hypha. The mass of hypha that form the body of the fungus is called mycelium. There are two major classes of mycorrhizal fungi, ectotrophic mycorrhiza and vesicular-arbuscular mycorrhiza, that is VAM fungi. So, we start with ectotrophic mycorrhizal fungi, which shows a thick sheath or mantle or fungal mycelium around the roots, and some of the mycelium penetrates between the cortical cells. The cortical cells themselves are not penetrated by the fungal hypha but instead are surrounded by a network of hypha called Hartig net. Often the amount of fungal mycelium is so extensive that its total mass is comparable to that of the roots themselves. The fungal mycelium also extends into the soil away from this compact mantle where it forms individual hypha or strands containing fruiting bodies. The capacity of the root system to absorb nutrients is improved by the presence of external fungal hypha that are much finer than plant roots and can reach beyond the area of nutrient depleted soil near the roots. Ectotrophic mycorrhizal fungi infect exclusively tree species of gymnosperm and woody angiosperm. The next one is vesicular-arbuscular mycorrhiza, that is VAM fungi, do not produce a compact mantle of fungal mycelium around the root. Instead, the hypha grow in the less dense arrangement both within the root itself and extending outward from the root into the surrounding soil. After entering the root through either the epidermis or a root hair, the hypha not only extend through the regions between cells but also penetrate individual cells of the cortex. Within the cell, the hypha can form oval structure called vesicles and branched structures called arbuscules. So, as the name suggests, vesicular-arbuscular fungi, that is because it forms oval structures, vesicles, and branch structures, arbuscules. The arbuscules appear to be the sites of nutrient transfer between fungus and the host plant. Here in this diagram, we can see here that how vesicles are found inside the cells and how arbuscules surrounds the cortical cells. And in this diagram, we can see here that how Hartig net and fungal sheath have completely arrested the root of any tree. So in this comparative diagram, we can see both type of, we can see the penetration module of both type of fungi. So association of vesicular-arbuscular mycorrhizal fungi with the section of plant root. The fungal hypha grow into the intercellular wall spaces of the cortex and penetrate individual cortical cells. Outside of the root, the external mycelium can extend several centimeters away from the root and may contain spore-bearing structures. Unlike the ectotrophic mycorrhiza, vesicular-arbuscular mycorrhiza make up only a small mass of fungal material, which is unlikely to exceed 10% of the root weight. Vesicular-arbuscular mycorrhiza are found in association with the roots of most species of herbaceous angiosperm. The association of VAM with plant roots facilitate the uptake of phosphorus and trace metals like zinc and copper. Calculation shows that root associated with mycorrhizal fungi can transport phosphate at a rate more than 4 times higher than that of the root not associated with mycorrhiza. The external mycelium of the ectotrophic mycorrhiza can also absorb phosphate and make it available to the plant. In addition, it has been suggested that ectotrophic mycorrhiza proliferate in the organic litter of the soil and hydrolyze organic phosphorus for transfer of the roots. The next topic is nutrients move from mycorrhizal fungi to the root cells. Little is known about the mechanism by which the mineral nutrients absorbed by mycorrhizal fungi are transferred to the cells of plant roots. With ectotrophic mycorrhiza, inorganic phosphate may simply diffuse from the hypha in the Hartig net and be absorbed by the root cortical cells. With vesicular-arbuscular mycorrhiza, the situation may be more complex. Nutrients may diffuse from intact arbuscules to root cortical cells. Alternatively, because more root arbuscules are continually degenerating while new ones are forming, degenerating arbuscules may release their internal content to the host root cells. सो यहाँ पे हम देखेंगे कि जो एक्टोट्रॉपिक माइकोराइजा है वो सिंपली डिफ्यूज करता है हाइफा और हार्टिग नेट से, जो भी उसने एब्जॉर्ब किया वो कॉर्टिकल सेल में. बट जो वैम है उसका सिचुएशन थोड़ा कॉम्प्लेक्स है. उसका जो उसने जो एब्जॉर्ब किया उसको डिफ्यूज़ तो करते ही हैं कॉर्टिकल सेल में, साथ ही साथ जो इसके वेसाइकल्स हैं या आर्बिस्क्यूल्स हैं वो कंटिन्यूअली डिजेनरेट भी होते रहते हैं और इसकी वजह से उनका जो इंटरनल मटेरियल है वो भी रूट सेल्स में जाता रहता है. A key factor in the context in the extent of mycorrhizal association with the plant root is the nutritional status of the host cell. तो न्यूट्रिशनल स्टेटस क्या है होस्ट सेल का, कि मतलब वो कितना डेफिशिएंट है, मॉडरेट डेफिशिएंसी है या हाई डेफिशिएंसी है या बिल्कुल डेफिशिएंट नहीं है. उसी बेसिस पे माइकोराइजल एसोसिएशन अपना ग्रोथ करते हैं. सो मॉडरेट डेफिशिएंसी ऑफ न्यूट्रिएंट सच एज फॉस्फोरस टेंड्स टू प्रमोट इन्फेक्शन, whereas plant with abundant nutrient tend to suppress mycorrhizal infection. मतलब जहाँ ज़रूरत नहीं है वहाँ पे वो सप्रेस करता है इन्फेक्शन को और जहाँ ज़रूरत है वहाँ पे वो इंक्रीज करता है. माइकोराइजल एसोसिएशन इन वेल फर्टिलाइज्ड सॉइल में शिफ्ट फ्रॉम ए symbiotic relation to parasitic one. In that fungus still obtain carbohydrate from the host plant, but but the host plant no longer benefited from the improved nutrient uptake efficiency. अगर ज़रूरत नहीं है या वेल फर्टिलाइज्ड है तो वहाँ पर सिर्फ माइकोराइजा होस्ट प्लांट से कार्बोहाइड्रेट लेगा और यहाँ पर वो सिम्बायोटिक एसोसिएशन के जगह पे पैरासिटिक एसोसिएशन शो करेगा. Under such conditions, the host plant may treat mycorrhizal fungi as it does the other pathogens from carbon dioxide, water and mineral elements. So, no, this point is from the summary of this chapter. So here we are ending with this topic, that the last point is, under such conditions the host plant may treat mycorrhizal fungi as it does with other pathogens. जब वेल फर्टिलाइज्ड सॉइल है और प्लांट्स को नो नीड है मिनरल्स की, तो उस वक्त माइकोराइजा पैरासाइट की तरफ वर्क करने लगते हैं. मतलब ये खुद की बेनिफिट तो लेते हैं, कार्बोहाइड्रेट एब्जॉर्ब करते हैं, बट दे डू नॉट गिव देयर फॉस्फोरस और एनी टाइप ऑफ मिनरल टू द होस्ट प्लांट. और उस कंडीशन में होस्ट प्लांट भी माइकोराइजा के साथ वैसे ही ट्रीट करते हैं जैसे कि वो कोई पैथोजेन हो. सो we have completed this chapter with great efficiency, with great enthusiasm, and we arrive with next chapter, which will be more interesting and more important with the point of view of botany or plant pathology or phytology itself. So thank you for listening me.